Good morning and welcome to Medicine Grand Rounds. It's really my pleasure this morning to introduce one of our own, Dr. Rozalyn Anderson. Dr. Anderson is a graduate of Trinity College in Dublin for her undergraduate degree and then took her PhD at University College Dublin. After a year as a junior lecturer at University College Dublin, she came to the United States where for 3 years she was a postdoctoral fellow in the Department of Pathology at Harvard, and then came to the University of Wisconsin in 2003 as a postdoctoral fellow at the Institute on Aging. She has served as a member of our faculty as an assistant professor, and in 2016 was promoted to associate professor with tenure. She also serves as a health officer for the Grec VA Hospital, as well as a member of the research committee for the VA. She's a member of ICTR and the Cancer Center, an affiliate member of the Mortgage Institute, the Alzheimer's Disease Research Center, the Institute on Aging, and our Division of Endocrinology, Diabetes, and Metabolism. She's published over 37 original manuscripts in very high-impact journals, including Nature, Science, BMC, Genomics, Aging, Cell, and Nature Communications, both twice in the last several years. She's well supported for her research, currently with an R01 from the NIA, an American Federation of Aging Research Glenn Foundation Award, for which she's also PI, and is also a co-investigator on another R01. She's performed extensive review service for the NIH, NIA, and currently is a member of the Career Development Award study section for the NIA. She serves as a mentor for the Interdepartmental Graduate Program in Nutritional Sciences, and here at the Institute on Aging, T32. Nationally, she's a fellow of the Gerontological Society of America and is a member of the American Aging Association, the American Society of Biochemistry and Molecular Biology. She's also been a generous citizen in terms of editorial duties. She's co-deputy editor for the Journals of Gerontological Biological Science section and is on the editorial board of 4 other journals. And she's received a number of awards along the way, including Paper of the Year Award from Aging Cell in 2008. She received the Nathan Schock New Investigator Award from the Gerontological Society of America, and in 2016 received the AFAR Glenn Foundation for Medical Research Breakthroughs in Gerontology Award. So once again, here's an example of a member of our own faculty, really of national and international stature. And for that reason, we're really privileged to have her give grand rounds today, entitled Elixir of Youth, a 21st Century Gerontological Quest. Please join me in welcoming Dr. Anderson.
Thanks, that's great. Thank you for that very kind and detailed introduction. Yikes. So this is my disclosure. Apparently I have to show that. Here it is. I have nothing to disclose. That reminds me of Oscar Wilde. Everyone knows nothing to declare except my genius. I wouldn't say as much myself, but it's one of my favorite lines. So this is the title, as you can see, Elixir of Youth, a 21st Century Gerontological Quest. I was told to come up with something snappy, so it's not short, but I think it's kind of capturing the idea of what my work is after. The Elixir of Youth, for a start, is there a way that we can delay aging? That's really the goal of the research, and so I'll be showing you a bunch of colorful data, different animal models, and some ideas here and there, but ultimately what we're interested in doing is finding a way to delay the pace of aging. That's the goal, and I'll tell you why in a few moments. This 21st century part is that things have transformed in the last really over the last decade in terms of what we can do to inquire about what's happening in the biology of aging, what contributes to aging, and in my hands, using the model of caloric restriction, how is it that delayed aging comes about? So using these very modern cutting-edge tools, we can start to peel back layers of what's going on to understand the very fundamental biology of what creates disease vulnerability and how we might go after that, targeting it pharmacologically and develop this elixir of youth. Okay, so I'm just going to show you this is a slide here because I always get carried away and then I have to go at double speed to thank everybody for all their great contributions. These are the people in my lab here. I've got collaborators within the School of Medicine and Public Health and, of course, the GREC, which generously supports me here. You may be familiar with some of these people in our department. I've got links to departments across campus. I just recently took up the position of Director of Metabolism of Aging program. What we're really trying to do is bring in people from all around campus to have aging as a focus in their work. And it's turned out to be a really fun ride, and only possible on this campus where we've got such marvelous faculty, marvelous expertise scattered across. So I encourage you to venture out from the west side onto campus and explore some of our colleagues' work. So these are listed here. We've got a bevy of biostatisticians. They went from being the people I just tolerated to now I'm in love with all of them. They are so necessary. And so we've got biostatisticians here at the department, also on campus, and then off-site in Alabama, because that's where the best ones are grown, apparently. I've got support here, and then some colleagues that I've published with in the last year that I wanted to acknowledge right there. So this is the lab team. This is us in our GREC area up in the VA, just upstairs. And you can see a great set of scholars, and I'm going to show you work from some of these people today, but not all. And just so that you're really convinced that we are on a quest to find the elixir of youth. That's them coming to work in the morning. Okay, so I'm just going to get to the meat of what it is that we do. So aging itself is the most significant risk factor for a range of diseases. We all know this. So the idea is that if we could go in after aging itself, that we could delay across the spectrum of chronic diseases that occur as a function of age, for which the risk is increased. So if we can think about this, the caloric restriction studies, this is a proven model of delayed aging. The animals are healthier and they live longer. So indices of morbidity occur later, and there are fewer of them. So this is a bona fide delay in aging. If we can understand how that works, it will inform us as to what are the signature events that are occurring during aging. The next thing I think I really want to convey is that non-human primates are a great model for aging research. And I know it's a bit of monkeying around. They're very expensive. They're hard to work with. They're hairy, they're scary, and they smell bad. But they're highly informative. And so mostly what I deal with are the Reese's Pieces, not the whole monkeys. I'm very afraid of them. But we bring them pieces of monkeys over to the lab here, and those are the data that I'm going to share with you today. So the idea here is that the mechanisms engaged by CR would be suitable targets for development of interventions. And so this is really the foundation of the work that we do. So why does CR matter? Well, look at all of these conditions that are impinged upon by caloric restriction. And these are data from monkeys and from mice and rats. And so the literature on CR is really going back for years. It was first reported in 1935. I'm sure there's no one in the room who was born at that time. So this is a multi-generation of careers engaged in caloric restriction research. And you can see cognition, sarcopenia, colon health, brain atrophy. We reported that ourselves in 2009 with our team with Sterling Johnson. Cancer, amazing cancer outcomes. Since 1944, we've known this. Diabetes, arthritis, and the whole works. So here's an intervention that impinges on all of those diseases simultaneously. This is just a quick look at what CR is and what we know about it. This would be considered the classic CR rodent study. And basically, you take groups of animals and you put them on different diets, shown here. As much as they like, 25% less, and this is a pretty hard, this is a heavy blow, 55% of their, what they would ordinarily prefer to eat. And as you can see, not surprisingly, they weigh less when they eat less. That was a revelation. What is a revelation, though, is this difference in their survival. So this is how many are alive, and this, when they're down here, they're all dead. And you can see this is the curve for the animals who eat as much as they want. The ones who eat less and the ones who eat quite a lot less have this huge lifespan extension. And if we map it this way, showing lifespan against calorie intake, you can see, importantly here, there's an increase in the average lifespan, which can happen with, hygiene, antibiotics, stuff like that. But there's an increase in the maximal lifespan. So even the longest lived are much longer lived than the controls, just simply by being put on this low-calorie diet. So we've published a bunch of things on this. There are reviews galore. If anyone's interested in finding out about it, please contact me and I'll point you away from the rubbishy ones. As you can imagine, nutrition and health is like a flea market on the internet. It's almost impossible to find anything credible. You can have anything you choose could be good for you, and scientists say, and anything could be bad for you too, something on the same page. So there are loads of great reviews, and there are loads of not so great reviews. So if you are interested, just give me a buzz and we'll set you up. But these are some of the factors. So on my other part of the lab, when we're working at mice in particular, we're looking at some of these factors here as mediators of CR. So the idea is it's not just that everything's happening slowly. The idea is that there's a program implemented that impinges on the rate of aging. So you think of it, nutrients in, sensed by factors, factors go and do what they do, and that creates the longevity program. So we call this an active response to CR. And we've shown this, some of the earliest work that I did in caloric restriction in yeast was among the first to demonstrate that there is an active reprogramming of metabolism as a function of caloric restriction. So these are the key factors, and I think you'll be familiar with some of them if you cast your minds back. Obviously, NAD is a critical factor in all redox. All of intermediary metabolism is touched on by NAD. And there are these sensor proteins, sirtuins, that are important in regulating protein function. I don't want to get into too much detail here, but we've got a series of sensors here that we know are responsive to CR. A series of factors here that have been implicated in aging using genetic means and in other species. And so these effectors here all produce this change in metabolism that we think is central to delayed aging. Okay, so we started digging into this, and I'm going to show you some data slides, but I don't want anyone to get too caught up on the details here. It's really broad strokes that I'd like to emphasize at this stage. In this study, we were kind of thinking, well, okay, so the animal ages all in one go. It's not like your ears stay young and your backside gets older. Everything happens together. It's all coordinated. But we wondered, so each tissue has its own function and it has its own signature. Is there an underlying signature that's responsive to CR across tissues? So can we find that shared kernel response that's independent of what type of tissue you're looking in? And it turns out that we could. And so these are all different tissues from mice, 8 different tissues from mice shown here, using gene expression data. And so what we did was we queried the data looking for pathways that were responsive to CR, and then asked if there was anything central to all of those 8 different tissues. So these are brain regions here, inner ear, heart, liver, kidney, adipose tissue, and skeletal muscle, gastrocnemius. And this is the signature that we found. So each of these tissues, even though they're wildly different in function, respond with this central conserved signature of CR, and it's massively involved in mitochondrial metabolism. So that's cool. We said, okay, that's the CR signature. Then we asked, well, is it only a CR signature in mice? And the answer is no, because we have these other species on CR right here, and they also produce the signature. And there's, well, so it's CR for sure, but is it longevity? So we asked in these genetic models of extended longevity. These animals have defects in growth hormone signaling axis, the somatotropic axis, and they live long. And they also, in part, produce the signature. It's shared, it's not exactly identical, but part of the signature are coming in. And these all were validated statistically. And so, okay, so now it's not just a signature of CR, it's a signature of longevity. Is it also a signature of health? And it turns out, yes, it is. So here are another set of transcriptional data from a variety of different studies where the animals, or persons, but we're animals too, are compromised. They have some metabolic compromise. And you can see that the pathways that are responding in these metabolically compromised animals and people is the inverse of our CR signature, right? And so then if you take studies where the cohort had a metabolic problem that was then fixed pharmacologically or clinically, you can see, look at this, the signature lights up again. So we have this signature of CR that's also a signature of longevity, that's also a signature of metabolic health status. So we thought that was pretty cool. And it kind of rolls into this idea here. So this is our working model. This thing's as old as all get out, but I haven't come up with anything, that's better than it. And this is maybe 12 years in the running now. But we have this idea here where there's a mitochondrial dysfunction underpinning a lot of poor health. We're seeing this in age-associated diseases across the board, this metabolic and mitochondrial component. Even diseases that have completely distinct etiology, when they manifest, there's this signature that's shared across in terms of mitochondrial function. So dietary excess, we know a lot from looking at individuals with these very rare disorders of the mitochondria, and also chronological aging. And this is to set it apart from biological aging. There's a whole thing in the field now where your biological age is younger than your chronological age. I think people think that it sounds fancy. We get some shirts made and parade around. My biological age is. And so this is the chronological age, morbidity and mortality. And here what CR is doing here is the altered mitochondrial metabolism that leads to longevity. So we were really encouraged by those transcriptional data. It was a completely unbiased approach, and thankfully, a few matched the model that we'd proposed some years earlier. So the next thing I want to talk about is the translatability of CR research. And for this, I want to introduce the monkey model and why the monkey model matters. And so I work in rhesus macaque, and they live maximum, their average lifespan is about 26 years of age, so that's a lot of per diems, if you can imagine. And they have a lot of the same kind of developmental passage that people have, in terms of, they're born very immature and incapable, and it takes them a while to get up to speed, although, they're engaged in romantic affairs a lot earlier than we are. That ×3 multiplying factor to get you from monkeys to humans doesn't count in that regard. But you can see they have a whole bunch of outcomes of development and age that are paralleled in humans. And they're a great model for human disease. And what I want to show here is, when you see old animals, you can tell right away they're old, but they have this same thing we have with fat mass, where it increases in middle age. I won't ask for a show of hands. It increases in middle age. It's very difficult to do anything about that. And then a slight decline at very old age. So fat mass changes with age. And then they have these great signatures of sag and bulge, and I won't ask for a show of hands there either. But this is the loss of muscle tone. So, this is frivolous enough, but we also see indices, cancer, diabetes, sarcopenia, all the things I showed you in that diagram earlier. The monkeys are a model for that. So these are monkeys from our caloric restriction and aging program here at UW. And the program's been going on for almost 30 years now. And what I'm showing you here are 2 animals that are the same age, okay? But the difference between them is one is a control and the other is CR. And I think it's pretty apparent the difference really here, this old geezer versus luxurious. Look at that hair. Don't you want to just... Yeah, enough with the cat pictures. We need monkey pictures on the web. ICANN has good hair. And so you can see there are overt differences in these animals, even though they're the same age, and that single thing driving it is the fact that they're on these 2 different diets. This is a review, or this isn't a review, it's primary data, but this is just old. This is published in 2009, and we were able to show this separation in terms of animals with age-related disease. So that's a huge gap there between the controls and the CRs, as you'll see, and it's almost a 3 times difference in the hazard ratio for disease incidence for age-related disease. This is huge. These are the actual individual diseases we reported at that time, and it was pretty clear and significant. Subsequently, we published again when we had the first pass, we had identified a difference in age-related mortality, but not all-cause. So all-cause mortality includes accidents or things that are not associated with aging per se, but we were able to show that both of these things were significant. You have to wait till the cohort gets old enough to be able to get these data. And so just to show you our more recent data, this is again, how many years later? 5 years later, we still see that separation between the controls and the CRs in terms of morbidity, and here are the incidences of the various disease. Our hazard ratio is holding, and we still have significance. And so it was an interesting thing, just for any of you like career-wise, we'll take a quick segue. And when we published our first study, we'd shown this effect on aging, and it was really neat. A parallel study at NIA, Intramural, had published their study where they didn't see a difference. And it made a great splash, and they had the CR doesn't work in primates. And we're like, oh, yes, it does. You just haven't looked at the data. And so instead of getting into a mudslinging operation, boy, was I tempted. Instead, we sat down together and came up with a way to resolve this by comparing the 2 studies side by side. And the result is this publication here, featuring the NIA people here, and there, and the UW people, and then there's our Alabama brethren right there. And so what we were able to do is look at both sets of data, and then we were able to discover that the reason they hadn't seen a difference between their controls and CRs is because all their animals were on CR, and they were comparing CR to CR. And in their old age cohort, they had animals that reached 40 years of age, which was the maximum ever recorded for this species in captivity. And they even had one animal that lived for 43 years of age, which is the oldest monkey ever. So the idea that there was no impact on aging is really a joke. But it really goes to show you a couple of things. The importance of your control. Who are you comparing it to? And then the importance of a diplomatic strategy, because we'd still be mudslinging if we hadn't gotten together as a group to resolve the controversy. Okay, so what does CR look like? This is a short, just after 2 years of intervention, but I wanted to show things that people might observe in a clinic at any old time, indices that you're interested in looking at. So there's a change in body weight. This is just showing 2 years of CR, and we have the controls here. Their transition goes up, and the CRs go down. So body weight changes, fat mass changes. One of the hallmarks of CR is this improvement in glucoregulatory function. Lower glucose, fasting glucose, lower insulin, and improved performance on the frequently sampled glucose tolerance test. So we see this across, this is always the thing with CR. So this nice, clean, whistling clean glucoregulatory function. So we see the glucose, insulin, and insulin sensitivity all affected by CR. So this is nice because these are things you might recognize in the clinic. But what really is cool is this human clinical trial of caloric restriction that was funded at great effort and ginormous expense by NIA. And in this study called Calorie, it's spelt wrong because I know all you guys in the medical profession like to have acronyms. And sometimes I think you just pick random letters from any collection of words and make it into a thing. I see them all the time at the Research and Development Committee. But this is an actual acronym. I can't remember what it is. All I know is it's a bad spelling of calorie. And so this was set up in 3 locations across the country, over 200 individuals. And here's the key. These were people who were not overweight. They were in the normal weight range. And they were also not old, 21 to 50. And 50 is not old, by the way, just by the way. And so this study was set up. And what was really sweet was they did all these measures. And so in human CR, 2 years of intervention, they tried to get 25% calorie reduction. I think they got more like 15, but it was in a spread. Lower body weight, lower adiposity, lower circulating glucose, insulin. Does this look familiar? Because I just showed you these data, didn't I? Yeah, so the monkeys have the same effect as the humans. And CR delays aging in monkeys. So the same outcomes, the same parameters, it looks very promising that the CR paradigm would actually, if someone were crazy enough to do it, delay aging and extend longevity in humans. There's every reason to believe those parallels will hold. Okay, so uncovering the mechanisms of CR. Now, don't be alarmed. I know people look at that and they go, oh, here we go. Actually, I have the same feeling myself. What I want to show you is just the different tools that we're applying now to try and understand at the molecular level what's happening in response to CR. What explains this just completely different trajectory of aging that we observe in the CR animals? So I'm showing you here transcriptional data. We do these alignments, We can do pathway analysis, which is nice. So you measure all the transcripts and you ask, what do they represent? What kind of pathways and processes do these transcripts that I detect in this piece of monkey liver, what do they represent? And this is the analysis we do here. And you can see a whole bunch of different metabolic pathways that are stimulated in response to CR, and a whole bunch of immune and inflammatory pathways that are attenuated in response to CR. So you have this push-pull, lower inflammatory tone, and a shift in metabolism, in metabolic pathways. So we were able to go further in there, look at individual genes. This is what the plots look like. And then we were able to identify this really peculiar thing that we hadn't realized, that a whole bunch of these transcripts are actually present as different isoforms. And you can't get that from the old standard microarray. And does anyone ever run a microarray experiment? Okay, never mind. In those experiments, you probe, you have to know what you kind of ask, is this here? Whereas in the RNA sequencing, instead, you get, this is here. You see the difference? So one is kind of targeted and the other is not. And we can see different transcripts being produced as a result of CR. I'm showing those ones with separate isoforms here. So we dug a little deeper, and it turns out there's a whole bunch of RNA processing of pathways that's specifically induced by CR. So these are just showing you the different exon changes in exon usage here. This is an exon map showing you where the differences arise between control and CR. And we see things like exon skipping or alternate exon inclusion. And so when we look at all of these data, look at this. So this is the important piece here. The control animals do not implement this mechanism. The CR animals do, right? So this is just a massive 60-fold change here in the engagement of this pathway. And these are the pathways, the cellular processes for which there are factors regulated in this manner. So this is the first demonstration of RNA processing being specifically engaged in CR in a mammal at all, let alone a monkey. So this kind of thing would have been just completely impossible even 15 years ago. So it's really neat. So we looked at the transcriptome, we saw metabolic pathways, we saw immune pathways. We looked at the proteome. So this is mass Spectator. What you can see here is a collection of all the proteins, all the peptides related, and we map them back to proteins here. And then you can produce maps like this that show you that the spliceosome is upregulated with CR. And that matches what I just showed you with the transcriptional data. So now we've got a completely different platform, completely different way of asking what the heck is going on. It's telling us the same message. It's telling us that we have metabolic pathways and that we have this RNA processing. So the next thing I want to show you is this acetylome. So this is, post-translational modification of proteins. So this is where you stick something onto the protein and it changes what it does, who it hangs out with, where it goes, and what it's capable of. It's really handy. And so acetylation is one of those kind of emergent modifications. And what's really interesting about it is it really changes in response to metabolism. So we see it changing with age, we see it changing with feeding and fasting, and we see it changing with exercise. So it's very dynamic. And we were able to show that there was huge induction of changes in acetylation, specifically with caloric restriction. These are all, look at all these metabolic pathways. They all have regulation by acetylation. Now, not everybody in every pathway, in every shape and every form, but each one has factors that are regulated by this mechanism. So you can see it's massively pervasive all the way across cellular metabolism. And so this is just to show you individual proteins in the mitochondria. As anyone, they've mapped the entire proteome of the mitochondria, and you'll never guess what they called it, the mitocarta. It's so funny. I think it's a bit grandiose. And I was talking to the guy whose paper it was the other day and mistakenly called it the Magna Carta. He said, "Are you trying to be funny?" I said, "Whoa." I thought you were when you called it the mitocarta. So you can see these massive changes here in the acetylome. So we put all these data together. We had, and this is, transcriptome, proteome, metabolome, lipidome, acetylome. Yeah, everything. There's a new thing now, have you heard of it? The exposome. That's everything all together. It's in the nutrition field, especially with the policymakers. And I was like, "What do you mean exposome?" We said, "Oh, well, it's just everything." I was like, "Why did you just call it everything?" So we put everything in the pot and did this giant regression with Sean McElwain, who's just a wonderful statistician and part of our bioinformatics and medical statistics. I can never get that right. There's a B and an M in there somewhere. The department that's affiliated with ICTR. And so we had this huge correlation plot. And what was really cool is that we were able to identify mega clusters of function. See these things that all moved together. And the map for CR was completely different from the map for controls. So we were able to show clusters of functional change, even considering all those different 20,000 molecules done in pairwise comparison. It took over 24 hours to run. Identified these clusters and this completely separate trajectory. So it's really beautiful to be able to look in this way. A completely separate way of looking at it is random forest. And so in this, I don't want to bore you, but basically it's a really cool computational technique where you put everything in the pot and you get it sorted into little, what are called trees. And then trees populate the forest, and you ask who in the forest can identify person group A from group B. So it's a completely unbiased approach. So we ran this for the proteome data, acetylome data, and transcriptome data. The proteome data looked like this and pull these things out. Totally different set of factors, but the same pathways. Wait till you see. We stuck the random forest gene, protein, and acetylome models all together, and while none of them really created a story by themselves, when put together, they made this huge story. This is a string map looking at connectivity among factors. I did twice the amount of connectivity that you'd expect by random chance. And then here's the doozy. Half of the things in this network are regulated by RNA processing. So it's really beautiful. So we've identified a brand new way to implement change across metabolism through RNA processing, and it's specifically brought on by caloric restriction. So here we are. This is our model again. Oh, I must have taken out the little, the groovy R. Oh, there it is, yeah. So that's what I've been showing you, right? This is the next part, and I'm not going to dwell on it. We just started working on this now. We have a paper that we're hoping to submit really soon, or the graduate student is going to turn inside out. I keep seeing him on campus, even though he's moved to San Diego. I go, "Oh, God, there's Carl again," because I have to finish the paper and send it in. I know. He's like appearing in my kitchen. He's not dead. So what he did this beautiful study. So we were asking, so if mitochondria are all that, we'll be able to show that by simply change mitochondria first and then exert some of the things we see with CR. So if mitochondria come first, as we think we do, then we should be able to reproduce that kind of CR-ish effect. And so we put this PGC1 alpha as a transcriptional coactivator. I won't go into it. Do you want to hear the name of it? It's peroxisome proliferator activated receptor gamma coactivator 1 alpha. I know, it's just ridiculous, isn't it? So PGC1 alpha, and we put it into cells, we exert a mitochondrial response, and we're able to show this massive change across multiple cell functions. NAD metabolism, we do this cool multiphoton stuff with Kevin Alcieri and Loci. If you have seen those, they're really beautiful. Growth indices, lipid composition, membrane composition, and cellular structure. And you look at the RNA-seq data, and you can see all these pathways, metabolism, immune, growth, structure, and homeostasis, all responding to the fact that we just changed their mitochondria. And we double-checked. It's not PGC1 alpha running amok with promiscuous gene activation. We checked. It's keeping its hands and arms inside the vehicle. This is an adaptive response to the change in the mitochondria. Again, we see this change involving shifts in exon usage, which we thought was also really beautiful. But the main signature here is this growth thing. So that's really neat because we had this as our model originally, and now what we're able to do is place this downstream of metabolism. So the animals are smaller with CR, right? All their parts are smaller, so you think growth. With a whole literature of growth deficient dwarf animals living longer. And so it had always been that these are two separate things. It turns out that the mitochondrial change is upstream of the growth inhibition, not downstream of the growth inhibition with caloric restriction. And so you can see it's in line with the established literature, but we're able to augment our pathway here. So the last thing I'm going to show very quickly, it's pretty much the last thing, is this new, so we were like, how is this going on? How are all these tissues talking to each other? How is it so coordinated? How are we getting that central response everywhere? How is it, and how is this coordinated across the animal? So we did this screen with collaborators. This is the guy from Brazil, and looking at microRNAs in circulation. Now, I was a skeptic. I was like, what the heck? RNA running around in blood. If anyone ever, okay, this is the olden days now, but if anyone's ever run a northern blot, you walk past it and your RNA goes away. So I was like highly skeptical. But of course, these have different structures and they're often protected, and we don't even know how they're transported about the place. In fact, we don't know where they come from and we don't know where they go. They're like the Willy Wonkas of systemic signaling. But we're getting some clues. And so we're able to sequence all of these from circulation from control and CR animals. And they separate it out nicely, where you can see here in this heat map and in this PCA map here. So 24 known and 10 novel microRNA change with CR. These are their predicted targets. Cancer, growth signaling, RNA processing, and metabolism. Look familiar to anyone? Isn't that cool? So the same things we identified as being mechanistically involved in CR are the putative targets of these microRNAs that we're detecting with CR for closing the circle. And so you can see different ways of mapping the data here. And what we're able to do is then get these synthetic versions of these and put them in cells and watch them impinge on growth regulatory functions and on cellular growth. So we know they're functional. And the next trick is going to be to identify where they come from. And I think we know. I think it's adipose tissue. This is showing you correlation of one in particular, MIR125A5P, and it correlates with body weight, adiposity, and insulin sensitivity, our hallmarks of CR. And this is the pathway that it impinges, that all of those CR responsive microRNA impinge upon, and it's the insulin signaling pathway. And there again is our hallmark of CR, improvement in insulin signaling and glucoregulatory function. So these are the key points. The beneficial effects of CR are closely linked to metabolism. There's just no two ways about it. I challenge anyone to a quest to find it otherwise. CR mechanisms are relevant to aging of long-lived species. I showed you the monkey data. I showed you the parallels with the human studies. I think it's just enormously exciting. I think what we find in the monkeys is going to translate very rapidly to humans and thereon to clinical application. There's a metabolic shift in liver. I've shown you that, and I don't want to go into too much detail about why that's important. RNA-based mechanisms are engaged in the response to CR, and that's just brand new stuff right there. It's really exciting. We think energy metabolism could be the driver in CR's mechanism. So it's not a passenger, it's not a consequence, it's actually pushing the entire program forward. And then we have microarray playing a role in homeostatic mechanisms. So what about the Elixir of Youth? This is a review that we published in eBiomedicine recently. That's a nice journal, by the way. It's a fusion of Cell Metabolism and The Lancet, and I'm on their advisory board. So please consider sending in your publications, reviews, perspectives. It's really taking off in a nice way. It's kind of both US and UK, which is really the way science is multinational, and so is the journal, and I think it's a nice eBiomedicine. So these are various factors that impinge on the pathways I've shown you as being mechanistically involved. PPAR agonists, they've fallen out of disfavor in the clinics, but they're still really useful tools to understand the primary biology, metabolic regulation, and how it impinges on inflammation. Resveratrol, you may have heard about that. If you drink enough wine, you should get enough of it. FGF21, which is a favorite of mine, but FGF21 extends lifespan in mice. It's really cool if you put in additional copies of it. Rapamycin also extends longevity in all the species it's been tested in, yeast, worms, flies, mice. They're doing it in dogs in Seattle, so we had such fun. The dogs in the street are on rapamycin, because the dogs in the street is an Irish phrase for everybody the heck, is doing something. So now finally the dogs in the street are on rapamycin. And then adiponectin, which is something we have a program we're building on right now. So these are the things that are going to need to be impinged upon to create the elixir, and these are the possible pharmacological applications. But you know what? It's too late, because apparently it's already available on the internet. Okay, I'm going to leave it there and take some questions. Thank you.
Thank you very much, Dr. Anderson. I'll ask you to call on the audience, and please repeat the question for the recording.
Righto. Yes?
I wanted to ask you a question about CR in relation to kidney function. I think, as you know, glomerular filtration rate decreases about 10% per decade after age 30. I wonder if there are any data regarding CR and the delay of decrease in GFR. What we see is some exceptions. Sometimes we find people in their 70s and 80s who don't have this expected decrease in GFR. I don't know if anybody's looked at any difference in there.
Okay, so this is a question about kidney function and the age-related. I'm not going to even try and say those things. I'm not a clinician, but there's disease of the kidney as a function of age. So this has been looked at in rodent models, I think rats primarily. So there is a literature on it. And I know that they've looked at, kidney structure and thickening of, I'm sorry, this sounds terrible, thickening of parts of the kidney. I never did anatomy. And so I don't really care about kidneys, but there is a literature and there is a delay in aging outcomes. There are some types of animals, some rodents that have kidneys that really die of kidney failure, and so I think that it's corrective in some of those regards. But to my knowledge, yes, it is impinging on kidney function. And I can point you to some of that literature if you like. Yeah. Sorry, Molly?
So another consistent biologic finding across aging is that the female in every species in rodents, primates, drosophila. How does that sex difference interact with or modify your function?
Okay, so this is a question about sex differences, sex dimorphism in longevity. This is known, yep, so across species, females tend to live longer than males when left undisturbed. And I think that we don't see so much of a big difference in mortality in our monkeys, but our monkeys are in captivity, so it's, I think if you looked at monkeys in the wild, you might see it. There's no difference in the response to CR. It's equally protective in males and females for us at this primate site here. And I think elsewhere in the NIA study, they didn't see a difference in the response to CR, so it was kind of hard to, their study was so screwed up. Different. And so in mice, I think there are different mice genetic backgrounds where you will see that sex dimorphism. And so the different genetic lines will respond differently. You see sex dimorphism in some, but by and large, if you're in hybrid lines, for example, you won't see it. There's also, the interventions testing program is an NIA-funded program to look for elixirs of youth. And they've looked at things like acarbose, 17-estradiol, rapamycin, metformin, things like that. And they see profound sex dimorphism among those agents they've identified as being able to impinge on longevity. It's really quite profound. So the sex thing is huge. Huge. Yes. I think there's a question over here.
There's been interest in extending the period of fasting between meals to promote autophagy. And are your calorically restricted mice, do they have more prolonged periods of time between eating? And is autophagy really, which is promoted enormously by no food intake at all for extended periods, is that playing a more central role in this, or is it really chronic hypocaloric?
Okay, so this is a question about periodic fasting and also its connection to autophagy. So it's really interesting. There's a whole field of people who are looking at intermittent fasting now as a therapy. There's beautiful work by Walter Longo looking at periods of, he uses a fasting mimicking diet because it's too tough for people to actually fast. And it improves resilience to things like surgery and chemotherapy. And he got beautiful data in mice, and it's translating very nicely to humans. Mark Matson is a guy at the NI Intramural, and he's also big proponent of the intermediate fasting. And there's some really nice, interesting data about how if you have extended fasting periods, it will attenuate the negative outcome of a lipotoxic diet, simply by limiting the time period of food. Now, the way our diet was implemented, they were all fasted overnight. There was no food from 4:30 in the afternoon onwards, and even then that was just a tiny snack. So they do have that fasting period. My mice are fed daily. There's no food left by 5 o'clock, so they're all fasting overnight as well. So even my control mice are not truly ad lib. I would never do that, because if you have ad lib, then they all get fat. And now you're measuring disease against caloric restriction instead of healthy, normal trajectory against calorie restriction. That's one of the differences across the literature. And so for the autophagy thing, yeah, you know, it's really hard to know. It's going to be tissue dependent. There's a slovenliness in the literature where you'll find that someone's done one Western blot or one, autophagy index assay or whatever, looking at, progression for the phagosomes in one tissue, in one animal, in one incidence, and they'll say, we've solved it in the abstract and the title. And so it's going to require a much more nuanced approach. We haven't looked at it in particular. I think where autophagy really comes into play is when you have these proteostatic defects, like clear defects, like neurodegenerative disease, for example, I think is going to be a big one, or some of these structural components. But we haven't looked at that specifically in our hands. Yeah.
Are you aware of anyone who's looked at total caloric intake in non-human primates who are allowed to eat all day long at will versus those that are fed episodically?
Well, that's interesting. So I was just talking to my colleague, Ricky Coleman, at the Primate Center about trying to do exactly something like that. It's very... Oh, sorry. This is about food intake and whether you could, if you set it up where animals had options to eat all through the day and how much they were eating versus this constrained, it's called in the literature time-restricted feeding. And it's something that we'd like to set up. But monkeys are the dickens. You know, they have these big pouches in their cheeks so they can, stash the stuff up there and retrieve it later. Handy if you're out on a hike. But it's precisely something that we're interested in. And we were just talking about that last week. It's a great idea. It's a good point. Is there someone here?
So if you come to the clinic and they've read this stuff online,
Yeah.
And they ask you what should I be doing for caloric restriction, is there a number that you go over? Is it cut in half, kind of like you're studying?
Well, so these animals were on 25% restriction. They were all measured at baseline. So we lowered them from their individual food intake. This wasn't just an applied, measured amount of food. It was adjusted individually for each animal. And we say it all the time. Don't even think about doing this without medical supervision. And there are people who do it, the cronies. So this is asking about people who want to go on CR. They always ask me at meetings what they should have for breakfast. I'm like, I don't care. But it's tricky because there's a balance there, and it's going to be qualified by your sex, by your age. Do you have comorbidities, and are you overweight? And all those things are going to be massively important. So I wouldn't recommend anyone do it. I know a bunch of people who do it. They're called the cronies, Calorie Restriction Optimal Nutrition Society. And some of them are, pretty normal looking, but others really aren't. They don't behave, they're always measuring themselves and, talking about it all the time. And so, it takes an unusual mindset to be able to do the CR program. I think it would work. I think it would, but I wouldn't do it. So I think all of this, it's a real problem across nutrition. So I'm also with nutritional sciences, is to know how to get, how to best inform people. You'd have to match their experience for them to be able to hear the message. It's like they say, oh, no one listens to nutritional information. They do. Look at this whole gluten business. Everyone's listening to that. They're buying all kinds of things, whether they have a gluten problem or not. So people do listen to the messages. It's just how they're pitched. So I wouldn't recommend anyone do that. But there is a very simple message, and I'm pretty sure all of you say this all the time. Try not to be overweight and get up off the couch. So it's abundantly clear. Just as much as CR is enhancing healthy aging, being overweight is the mirror opposite, and the same pathways are involved. It's just flipped. It's almost like a form of accelerated aging. Mm-hmm. The pink?
Yes, I have a question about the downsides, sort of segues with my last question. The downsides of caloric restriction, wartime malnutrition and increased death rates among people in Europe, for example, in World War II. Anorexia nervosa, and Lloyd Walford, one of the pioneers of caloric restriction, died of ALS at 79, which is not a really advanced age by our standards nowadays. And there was some question as to whether caloric restriction played a role in his disease environment.
So this is a question about the negative effects of caloric restriction, and it brings up some good points. I think Roy did a lot of things in his life. So he was a traveling Wilbury, taking temperatures of gurus on mountains in India, and he was just body rectal temperatures. He was an unusual fellow, and his biosphere experience, I think he ended up very malnourished after this. If everyone's heard of the biosphere, they all went into a bubble for 2 years, self-sustaining. They grew their own vegetables, filtered their own water, all this stuff. Really amazing. It's like in the 80s. And the idea was that if you were going to colonize another planet, you'd have to be able to be self-sustaining. And they lasted for quite a while, but they inadvertently all ended up being on CR. But not even really CR, there was a malnourishment. So the big point here is that there's a difference between caloric restriction and malnourishment, because that curve of extending your lifespan starts to dip back down again pretty quickly once you tip into malnutrition. Our monkeys were enriched for minerals and vitamins. And that's to bring them back up to recommended daily allowance. Although recommended daily allowance is another hornet's nest that no one wants to kick. It's all made up, if you ask me. So there's the anorexia nervosa, and there you have, I think, also a pretty clear mental health issues. And we have no idea how mental health intersects with physical function. We know that stress can produce these just dreadful physical outcomes, and we don't know the impact of repeated measures of those. Now, there is a model of small stresses that make you more resilient. It's called hormesis, and it's true, and it really works, but it's a very low-intensity stress. The idea is that it kind of kicks in your survival and resilience pathways by this small little perturbation in advance, and that's been shown in everything from people to plants. That stuff works, but a repeated stressor, a psychological stressor, I don't think that's going to be good for anyone anyway. But the bottom line is that CR, you can't have it's undernutrition, not malnutrition. Yeah.
You got a few minutes.
Oh, okay. I made the talk short so we would have time to talk. Yes?
In the study looking at humans being under caloric
Yeah.
restriction, did they look at how they were feeling?
Oh, they did. Oh, yeah, they did, yeah. And I met the people who do that stuff. I should really be less dismissive than that. Yes, they looked at... so this is a question about the humans. I'm glad you're there because I'm making a bags of this. I just get so... so this is the caloric restriction intervention, the Calorie Trial. And so that came out, this coordinating centre was in Duke, and it was all about the well-being, all about how do you feel, how does your cat feel, how did you feel when you left the house? And I was like, God. But they really wanted to know if people would tolerate it, and they did. they looked at things like sex drive and stuff like that. And they had this huge list of behavioral and feelings, I'm like, there's no feelings in science. But they did. They were very careful to look at things like that, and sleep disturbance, and any changes in patterns of behavior. And the whole, they just went on and on about it. It was totally safe to do and well tolerated. Not down at the 25% restriction, but at the 15, it looked great. And at the people who were on it looked great. So that was, it's really, there's not a negative downturn that way. But I can tell you from the cronies, there is a subset of them that are quite obsessed. There's a question behind there. Yeah. Okay. So this is a question about what happens with exercise and whether it's been combined with caloric restriction. And the answer is yes. It's even been combined with caloric restriction in humans. The Pennington Research Center in Baton Rouge, Louisiana, in the first phase of Calorie, they asked if they did a 25% calorie restriction and then compared that to 12.5% restriction and 12.5% increase in energetic output through exercise. So they did look at that, and the trajectories are different. So in humans, people. Mm-hmm. And so those data, Eric Ravson is the senior author. It's out of Pennington. You can find it on PubMed. They wrote loads of articles. And so it's not the same. Now, there are long history of exercise physiology. John Hollisey at St. Louis has been a pioneer in a lot of this work. He's really, truly amazing. He's still alive, and he's in his 90s and still working. That is old. 50 is not old. 90 is old. Okay. And so there's in those studies, it's pretty clear that you can improve certain elements of health through exercise. And I think we know this anecdotally in our own experience, but it doesn't impinge on aging per se. It doesn't change maximal lifespan. Okay, so you can improve health indices. And I think there's a critical difference between the leanness of an athlete and the leanness of a CR individual. The leanness of the athlete is still taking in huge amounts of energy and using it. So the pathways that are being engaged are not the same as the individual who's taking in less energy in the first place. What happens in the CR is basically a total reversion to fat metabolism. Everything's fat. They get these huge ketone bodies, lower respiratory exchange ratio, and they mobilize their fats super fast. So that loss in adiposity isn't just that their fats are smaller, their adipose tissue is different. And that's not the same thing that happens with the people on exercise. But exercise is a good thing. Yeah, 100%. Yes, a question over here.
Yes, there's been interest in metformin,
yeah.
potential modulator of the aging process. You didn't really mention that. I'm just curious about how.
Yes, okay, so it was on the last slide. This is a question about metformin as a means to, impinge on the processes that we're talking about here. So there is a human trial that's just started now called the TAME trial. Don't know what that stands for either. Trial of Aging and Metformin something. And so that's set out of, it's based up in Einstein in New York. And the idea is that they're going to be able to put people on metformin who are already older-aged individuals, and if the prediction is correct, the people on metformin will progress to fewer coincident morbidities than the people who are not. And this is the very first trial where aging itself is the target of the intervention. Up until now, and if this is successful, that will change the mind of the FDA. That may open the door for using this. Because the whole impinging on aging thing has been in the quackery arena. This supplement the tooth of something that came off something else while standing on one leg, and it will improve the glow of your teeth. Just weird stuff. So without FDA channels open, it won't be possible to develop these pharmaceuticals. There's a really interesting new development targeting cellular senescence with these so-called senolytics. It's really beautiful work coming out of the Mayo Clinic with Jim Kirkland. And obviously, the goal will be eventually to be able to use those in a kind of a clinical setting to actually target aging. So there's a lot riding on the success of the TAME trial, and metformin was chosen because it's most likely to be corrective of metabolic problems, even though it has not been shown to extend lifespan. So it's not going to change aging, but it could change health, whereas Resveratrol would fall into the same bag. Yes.
One more question.
Last one.
In your mice and monkeys, what provisions did you have to make to prevent malnutrition?
So this is asking about malnutrition. So we're lucky enough that there's, 1935 was the first study conducted. We never go anywhere near that 55% of normal intake. It's not necessary. You get massive extension, but it's not necessary. So we monitor, the animals are weighed weekly. The system is down. We know exactly. So I trained with Rick Weindruck, who was here, and he trained with Roy Walford, who was in the bubble. And so these are, he's like my granddaddy. And so there's a very firm set of protocols set up. And when the monkey study was being designed, it was informed by Weindruck and Walford's study design here at this institution. And that's why the monkeys were measured at baseline for their food intake and adjusted down from there. Yeah.
You have to supplement their diets to avoid malnutrition.
Yeah, so when we do a stringent calorie restriction, we use a slightly different diet that is enriched for minerals and vitamins for both the mice and the monkeys. The monkeys only have that enrichment. For the mice, if you go below 25% CR, we have a different diet. We try to match the calories so that they're not missing an essential nutrients. But yeah, it gets very tricky once you start messing around with the diet composition, because now, you've created all kinds of changes, and especially if the indices you're measuring are like and I didn't show you the metabolomic and lipidomic data, but if you have differences in the ratio of lipids, or even the composition of lipids, let alone the amount of fat in your diet, you can see that havoc being wrought throughout your data, and so it becomes difficult to assign coincident from causal changes. So we pay very close attention to that. Yeah.
So with that, we're going to have to close. I want to thank Dr. Anderson for an outstanding webinar.